Hey, Dr. Stansel, it's Ashley Allen. Today is September 23rd, 2018. This is microbiology, week 4, lab video, lab 1, video 2. This is on Methyl Red, Voges Proskauer and Catalase Testing. And I'm always going to apologize for my pronunciation. So at this point, we have already allowed our broth and the MVRP incubate for 48 hours. Now I've taken them out and I've split them up. So we have 2 tubes of E. coli and 2 tubes of S. epidermidis. So now we're actually going to expose these 2 samples to methyl red. Well, what's the point of that? So we know methyl red is a type of biochemical testing, which identifies various metabolic pathways of different bacteria. So methyl red is a pH indicator, basically. There are cultures that demonstrate mixed acid fermentation, and basically it does this without oxygen, so anaerobically. So when we add the methyl red, we're going to see what color it turns, right? That's going to indicate to us how acidic our sample is. And then I'm going to use the Barrett's A, which is for the Voges and Proskauer testing. That one we have to let sit for 30 minutes. So I'll just take pictures of that for you. But what that does is it identifies microbes that ferment glucose by the butanol pathway. So we're going to add 12 drops of Barrett's A into the other 2 tubes, wait 30 minutes, see what colors they come up with. A yellow is a more base, not very acidic, but sometimes Barrett regions can turn red as well, or a pink color, which just shows like an intermediate fermentation. So anyways, let me put on my protective gear and I'm going to turn the camera down so you can at least watch me use the methyl red and see what color our samples turn out. Okay, so I'm going to pull this over here so you can kind of see what's going on. So here's our sample. So here's methyl red. Okay, we're going to just cut the end and we'll disinfect all of this later. And remember, with the methyl red, we're placing 7 drops. So here's our S. epidermidis, and we're going to do 7 drops. So there's one on the end already. 1, 2, 3, 4, 5, 6, 7. So, oh my goodness, look at that color. That's like a beautiful red. So what does that tell you? Wow, that's beautiful. So that's S. epidermidis. Look how it doesn't really mix, which is interesting to me. I'll have to ask you about that one. And then here's our E. coli. Let's do 7 in there. 1, 2, 3, 4, 5, 6, 7. All right. And looky there. That one's also red. Notice the difference between E. coli and our S. epidermidis and how it's even mixing. So they're both red. They're both acidic byproducts. But look how this one's not really mixing and this one is. I mean, that just indicates 2 different bacteria, I'm sure. Way more out of my scope. But they're both red. They're both acidic. And next up, we're going to do the Voges Proskauer. And then we'll do the catalase testing. And I'll take some pictures for you. Thanks, Dr. Stansel.